Hi, I'm Princess Senate, conference producer at Technology Networks, and I am here to introduce our next talk, Multiomics Analysis of Gut Organoids as a Novel Tool to Model Diseases and Facilitate Precision Medicine. Please join me in welcoming Dr. Tamas Korcsmaros as your next speaker. Dr. Korcsmaros is a renowned researcher whose early work on the signaling database established him as a key figure in the field of bioinformatics. Driven by a multidisciplinary approach, Dr. Korcsmaros established a research group that combines computational and experimental methods, including the use of gut organoids. So Dr. Korcsmaros' expertise extends beyond academia with multiple innovation and industrial partnerships focused on developing new tools for analyzing complex data. A warm welcome to you, Tamas. Following the presentation, we will have a Q&A session and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. And without further delay, I will now hand over to Tamas to begin his presentation.
Thank you. Hello, everybody. It's really nice to be here. And hopefully I will be able to present you some interesting things on this topic. First of all, thank you very much for the introduction. A bit more information on my group, which we're currently work at the Imperial College London. We work lots of omics data sets, which we generate or we access from the public domain. And with different computational approaches, we're trying to interpret them and visualize them, and then we go back to the lab and do validations. My group's main interest is basically understanding cell-cell and cell microbe interactions related to gut homeostasis and precision medicine. And one of the diseases that we are mostly focusing on is inflammatory bowel disease. And as Princess mentioned, basically, I have been working with signaling networks for more than 15 years now. And we are particularly interested about how we can use organoids and to understand better host microbe interactions. So before I really go into the details, these are the main characters behind all this story. My great group, a very multidisciplinary bunch of people. Really like working with them, and they are very talented and working from the computer to the lab to the clinic. So in the previous talks, you have already heard about organoids. So this is just more like a summary on how we can generate organoids. There are ways to come from a embryonic pluripotent stem cell stage or from adults. You can take basically any kind of biopsy cell samples and you can induce pluripotent stem cells, or you can have an adult tissue biopsy and then you grow tissue organ-specific organoids that mostly I'm going to talk about, actually. I just wanted to highlight that for those people who are interested about organoids, but they don't have cell biology background or organoid background at all, we recently, with our colleagues, we wrote this review. So it's an open access review, and I suggest that this could be a good start, probably, for lots of people. Organoids are really great to grow different organ models. And then for why they are great, I'm going to discuss in the next couple of slides. They have already been used for different studies, as you have seen in the previous talks. And then probably there will be even more applications of organoids in the future. So before we go into the details, why do we need organoids at all? One of the reasons why it's relevant is that in general, we would like to investigate human tissues and human cells to understand diseases and health better, but it's often difficult to in vitro do that. And one option that for decades in the lab we have been using is immortalized cell lines, which are very easy in a way to in vitro cultivate. They are growing very nicely in most of the cases, and they have basic general functions. And there are lots of data in the literature. So if you have a data, then you can compare it, which is great. However, many of them are either cancer-related or being modified, immortalized, being kept in the lab cultures for years or decades. And usually they are kind of homogeneous in terms of cell types. The other scale is using animal models, which could be also great. They are very diverse. They could be very nice for testing complex systems. So in vivo studies from that point of view could be very useful. Longitudinal studies could be very nice. The issue is that they are expensive. Not all of them, or most of them, are not good for high throughput screening. Maybe C. elegans could be great for that. Some cases, some zebrafish works. And most of them have poor reflections on complex human physiology. Basic things, conserved processes can be nicely analyzed with them, but even mouse, as we started to know more and more recently, are not the best models for many things. So this is another summary from a really nice review about the different experimental model systems and what you can get out from them. So in terms of ease, maintenance, developmental biology related things, and basically how long experiments you can do with them, whether we can manipulate it, and all these things at cost, which is very important as well. So these are the traditional models, including modified mouse models. And then here come human organoids. So human organoids, as you can see, there are lots of green ticks, which is great. You can see that C. elegans and Drosophila has similar amount of ticks. But the biggest difference is obviously the physiological complexity, where it's much minimal compared to these organs. However, there's a really big green one here, which is recapitulation of human physiology. So that could be actually very useful. You can consider human organoids as kind of a great compromise in these features between the invertebrate models and the more complex vertebrate models that are not as good for certain points than for like human organoids could be. So with pros with organoids is that they can they could be their functions could be and structures could be very close to in vivo phenotypes. You have lots of cell types there. Epigenetics point of view, they preserve more the original tissue, and more importantly, from a medical point of view, they can be patient specific. So we are not only talking about often general models, but we can investigate a specific patient with that, and that could be very useful. And it was presented in the first talk. High throughput options are also there, although not always, as I will mention later, and they can be kept for a while. They could be really good for tissue-specific analysis, potentially host microbe interactions, drug screening. There are lots of things where they could be really good. However, and this is the slide, or this is the points when, in many cases, especially from companies working with organoids, you don't always hear, is that they are still very difficult to work with. One of the issues is standardization. We would like to compare conditions and patients, and for that we need really standardized protocols, and it's not always possible. Throughput can be high throughput, medium throughput, but in most of the labs it's not that high throughput. Disease-related organoids, for example, inflammatory bowel disease-related organoids have usually lower viability, while cancer organoids have a really nice viability. Complex co-culture models are still challenging, although very exciting. And working with multi-omics, which mostly I'm doing and then going to present, could be challenging because of the small sample numbers and size, but actually more and more possible. And in many cases, you need organoids and organoid lines or samples where you have also clinical metadata with them. And organoid biobanks could be very helpful for that, but there are not a lot of them yet. And the cost is very important. So one of the biggest bottleneck, I think, accessing organoid technology is the actual cost. Not always the setting up the laboratory, but basically maintaining an organoid culture. So that could be still a problem. So now I would like to more focus on my area on gut organoids and then why it's useful to use gut organoids. So the gastrointestinal tract, GI tract, is a really special place of our body, as you know, and there are lots of really interesting cell types there that are making sure that what we are eating is properly absorbed, and the microbiome content of our gut is basically well maintained. We are trying to keep the good bugs there and the bad bugs not. And cells like Paneth cells and goblet cells are very important in that. So investigating these cell types could be very challenging. And organoids could be really great for that. And the way how it works is, in a nutshell, is basically that you take a single intestinal stem cell or an intestinal crypt, and you can grow really nice organoids from them that contain lots of these cell types. Many of these cell types, like Paneth cells or goblet cells, can't really kept in cell lines and cell culture. So basically, it was nearly a big breakthrough opportunity to work with these cells, these human cells. Previously, like our lab and others, investigated Paneth cells before, but mostly from mouse. But now we can work with human Paneth cells, and they work differently. So that's really great. So this is just a short protocol for those who are more working in the lab, how you actually grow organoids. Basically, you get a tissue sample. It can, in the case of mouse, it's a whole intestine usually, but it can be a small biopsy. And then basically you do a scrapping, and then work on the crypt fraction to isolate it, and then you put it into an in vitro culture with different media that contains different growth factors, and then basically these stem cells starts growing and then later on differentiating. Basically, in 7 days, you can have this really nice, more complex structure. So, as I mentioned, for gut organoids, basically, you have the intestine with all these cell types. Take a biopsy, and then in 10 days you have this organoid. What we need really important to understand is that the intestinal epithelium contains these epithelial cells and many other cells in the mucosal layer, and then microbes. That's the intestine. An intestinal organoid, however, contains only epithelial cells. And during the process, you are basically eliminating anything else. So you will not have microbes, you will not have immune cells, fibroblasts, or other cells in a normal biopsy-derived process. If it's an iPSC-derived gut organoid, then you would have some mesenchymal cells, but let's keep this adult organoid-based process here. So on one side, that's great because you can work with these cells. But if you're interested, for example, about so that's really good for cell-cell interaction studies. You can add different cytokines or even co-culture some immune cells with these intestinal organoids. That's good. But if you're interested about cell microbe interactions, then it could be a bit problematic because the microbes should be in the middle. So one option is that there's a really nice protocol to make apical out organoids, basically a flip, where the basolateral side will be inside and the luminal side will be, the apical side will be out. In that case, you can put bacterial metabolites, for example, or bacterial molecules to the media and test their effect. Co-culturing bacteria with organoids is still challenging, mostly because of the aerobic condition that organoids like, and many bacteria doesn't really. But it's still a good first type of approach. And then there are more complex ones. So I already mentioned this apical out organoid one. Then there is this micro-injection option for a normal organoid, which could work, but requires an infrastructure. Sometimes it can be actually high throughput, but still there are certain cases when it's not the best way to do that. What is more trending is working with monolayers. So basically disrupting your 3D organoid and creating a 2D layer from them. And that works really well in many cases. And it allows a transfer to work with a transfer system. And here you can basically model the gut by having an upper part, luminal side, and then the bottom chamber for the basolateral side. And then basically in theory, and in many cases in practice, you can investigate cell microbe and cell-cell interactions in the same system. Recently, more and more complex systems providing scaffolding for these epithelial organoids, organ monolayers have arrived, and they provide flow and shear stress, and because of that, the organoid epithelium is actually better differentiated. They are even better than normal 3D organoids. And some of these gut and chip systems allows co-culturing with immune cells or other cells, even microbes. And there are even more complex systems, gut and chip systems like the Humix, developed in Paul Williams' lab in Luxembourg, for example, where there's a membrane there, and you can have an anaerobic condition on the upper side. You can have your organoids in the middle, and a great condition for neuronal cells, immune cells, or fibroblasts in the bottom chamber. So it's a very complex system, basically providing the environment for all of the cell types what they want. The only thing is that there's a membrane here, so molecules can go through, but there is no direct interaction there. If we plot these different models, then you can see that there's a throughput versus complexity comparison, and based on that, you can see that there are some complex ones, while there are some less complex ones, and obviously we are trying to go there. It's basically the issue is really the complexity and whether we can really have a throughput there. If you are interested about these models, a couple of years ago we wrote this review, which contains more information on details and examples of these models. So I mentioned at the beginning that organoids could be really great in precision medicine and also for omics studies. So let's see how they are. So this is very important for disease modeling. When I say that we can take a biopsy from a patient, we can isolate stem cells, grow organoids, but we can isolate other things. And we can isolate immune cells, we can isolate microbes. And we are working on projects where with collaborators where we are actually trying to do it from the same biopsy. And then we can do in vitro modeling of the same patients, same cells. And this kind of autologous testing is really, really interesting because it removes lots of external factors. The other very important aspect of disease modeling is the role of the different genetic backgrounds, as in previous talks were mentioned. And for this one, CRISPR modification, gene editing of organoids, could be one option that could be interesting. The other one, and I'm going to mention an example for that later, is that you can actually change the differentiation in organoids, and you can enrich the organoids for certain cell types. So you can modify that and then look at specific cell dysfunctions characteristic for certain diseases. And in terms of omics data generation, basically different omics approaches for host side, transcriptomics, proteomics, and then also for the microbial side, different metagenomics, metabolomics approaches, or metaproteomics actually, could be applied, and either directly on the organoid experiment, or you do it in the biopsy, and then you try to recapitulate it from organoids, and then the data from organoids could explain better in better resolution what you have seen in situ. Single cell approaches are getting more and more relevant with organoids, and there's the expensive way, you just do a single cell sequencing of your organoid culture, or there is a more cost-effective way when basically you are sorting your cells, effect sorting your cells, and then you do batch sequencing of those sorted cell types. And that, according to our experience, actually, that could result in really good results, and is much cheaper. So I think that the thing that more and more we will hear about organoids in the next 5 years will be screening, how we can use organoids for screening, as it was mentioned before today. And if you want to kind of structure this, I think there is these two types of screening. One is the testing many to one. So basically you use the same well-characterized organoid culture, or maybe you have 3 biological replicates for that. But basically they have kind of a fixed genotype. And then you expose it with different metabolites or microbes or drug libraries. And then you capture differences. I'm going to mention how you can capture that. The other option is the one-to-many, and this is more the precision medicine type of personalized approaches, when you already selected some useful compounds and you are interested about how they are, how different donors, different patients, different organoids could respond to that. It can come actually from the same patient, and you introduce different genetic changes, like a CRISPR screen of organoids with the same molecule tested on them. And I think it will be really powerful. Again, for both of these approaches, standardization is very, very important, especially for the one-to-many one. So I already mentioned basically the question of readout and what you can capture. And one of the things that I already said in the previous slide is it was omics, different omics readouts. The other one is probably more cost effective, is imaging. And I think organoid imaging is a really, really cool approach that can contribute to omics, not only to make it more cost effective, but also to increase the throughput. So we have an ongoing project on introducing different fluorescent reporters to organoids, in this case, an autophagy reporter, a process we are particularly interested, and then test these organoids with different bifidobacteria strains or other molecules. And with high throughput live cell imaging, we are trying to set up. We are investigating which cells are responding, which cells have an autophagy going up or down according to the different strains. And these two postdocs in our group who are working on that. So now I would like to go to a more specific examples after this introductory part, how organoids can be used for a new tool to understand particular disease. So as I mentioned, our group is mostly working with inflammatory bowel disease. IBD is a very interesting complex disease. Unfortunately, affects lots of people in a growing number, and it's a chronic lifelong disease. By definition, it's a dysregulation of the immune system, which is a connection to the changed microbiome, which is probably diet related, and there is some genetic associations to IBD. It feels very precise, immune system, microbiome, genetics, but actually how the combination is working and then why certain people develop IBD, certain not, and what age they do that, we don't really know. So there are lots of questions here. And both for Crohn's disease and ulcerative colitis, the two major types of IBD, patient stratification is a big issue. So basically, we don't really know which therapy would work best for a patient. And the best therapy has around a 30% success rate. For all the other patients have frequent relapses, fatigue, different digestion problems, psychological problems, and around 20% of them can't really work. So it's very important to understand first the basic biology and then how we can support therapeutic approaches. From a biological point of view, and why organoids could be relevant, if you look at the colon where ulcerative colitis is happening, for example, in a normal case, what you see is that there is this gut epithelium, really nice cells next to each other. Goblet cells are producing the mucus layer, so it divides the luminal part where the microbes and the different dietary compounds are. And then you have really nice surveilling immune cells. In an inflammation setting in ulcerative colitis, it completely changes the picture. You have leaks. Things can go into the mucosal layer, submucosal layer often. You have much less mucus because of less goblet cells or less functioning goblet cells. And you have very angry, overactivated different immune cells, inflammatory immune cells. Key thing, there's a major change in cell-cell and cell microbe interactions in IBD. So our focus is to investigate these cell-cell and cell microbe changes and how they are relevant for homeostasis. We are particularly focusing on goblet cells that are producing this mucus layer, mucin, Paneth cells that I mentioned earlier that are basically regulating the microbiome with antimicrobial peptides, and we are interested about different probiotics like bifidobacterium. So if we look at an IBD patient in a normal situation, goblet cells, Paneth cells, bifidobacteria, and other probiotics are working really well, and the system is fine. But and autophagy is very relevant for this process. But in certain cases, as I mentioned, this is not the case. In ulcerative colitis, goblet cells are not working properly. In Crohn's disease, Paneth cells are not working properly. In many cases, it is autophagy related. We have much less, these patients have much less probiotics in their gut. So there's a flare coming up. And different immune therapies are now there to achieve remission. And it's possible for 70% of the patients. But as I mentioned, at the end, half of them will relapse. So only 30% will have a constant remission or long-term remission. What we would like to do and understand is that how we can keep more patient in remission after removing the big inflammation, how we can make sure that it's not happening again. And this roller coaster, literal roller coaster, unfortunately, is not happening in the gut. And for that, we would like to understand basically these cells and their interaction better. So one of the first projects that we did was a systems genomics project when we looked at Paneth cells and goblet cells. So as I mentioned before, Paneth cells and goblet cells were difficult to investigate, and organoids help with that. And in the normal organoid, you have 10-15% of the cells are Paneth cells, and I think 20-30% are goblet cells, and then you have lots of enterocytes. What we did is that we used the protocol to enrich the organoids to have mostly 70-80% Paneth cells or 70-80% goblet cells. So basically, one organoid culture allowed us to focus on Paneth cells or focus on goblet cells. And after this, then we isolated the RNA from these organoids and did the sequencing. I said this sentence, I think, in less than 5 seconds. This was a 2-year part. So this box was a 2-year thing. There was not a lot of RNA normally extracted from these very small cultures. So we worked a lot on improving that. Nowadays, it's actually getting easier. But it was a really challenging and interesting project to make sure that we have enough RNA for sequencing. And at the end, I have to say that we overdone ourselves. We had 4 times more RNA that was needed. So we could do a very nice deep sequencing, focusing on microRNAs as well, which made the project even more exciting. And then after differential expression analysis of the transcriptomic data and combining it with different regulatory network resources, we were able to create the first regulatory network for Paneth cells and goblet cells. And then we did different types of computational analysis on them. I'm not going to really the details to that, but the key things were basically looking for key regulators who are responsible for Paneth cells and goblet cell differentiation. And then we compared these key regulators to known IBD genes that are associated to IBD, or processes associated to IBD. And it was really interesting to point out that there are lots of IBD-associated genes who did not have direct functions, why they are relevant in IBD, but we could point out that they could be relevant for goblet cell differentiation or Paneth cell differentiation. Then we had another project, which was actually a mouse-based organoid work. Here we were looking at the role of autophagy in Paneth cells. And in this case, we had access to a transgenic mice, which only in the intestinal epithelial cells, it was autophagy impaired. One of their key autophagy gene was not working. And then there was the control wild-type mouse. And basically, we grew Paneth cell enriched organoids from these. And then did a multiomics analysis. So both transcriptomics and proteomics from these organoids. And mainly we focused actually on the proteomics side. We were able to do a very, very interesting study showing why autophagy is essential in Paneth cells. And we identified lots of things that were not known before, connections, and relevant for Crohn's disease and this therapy. But we also had the transcriptomic data, and that was actually very useful for validation purposes. We learned also lots of things from that as well, but mostly we had a really strong findings on the proteomics, and we were able to publish this because we had the transcriptomics there as a control to validate that lots of things are happening on the proteomics protein level. And then going more in terms of systems medicine side, I would like to show how we can use organoids as a validation and data source for analysis of inflammatory bowel disease. So lots of work that I'm doing is related to networks. Networks when we are connecting 2 proteins, for example, or 2 genes. And basically, we previously developed lots of resources for this. So this is very computational work, systems biology work. We reconstructed the cellular signaling network for human and actually for other model organisms as well. And one of the resources, I'm going to show some of the resources we did. One of them was related to autophagy. And we were interested about how ATG16L1, a major autophagy gene, is implicated in Crohn's disease, is being regulated. And we created actually a comprehensive autophagy resource called AutophagyNet. And then this project I mentioned before, we combined the proteomics and transcriptomics data to validate the role of ATG16L1. We also created signaling, which is kind of one of the top signaling network resources. Very, very comprehensive resource if you're interested about signaling pathways in human. And this was the resource that we used for this study I mentioned in the previous slide on creating the cell type specific networks. So these cells obviously are receiving signals from other cells, mostly in the form of cytokines. And we created this resource called Cytokine Link. And with that, you can basically map cytokine-based interactions. And with the group of Nick Powell, we did a really, really interesting study when we exposed cytokines to different organoids, different cytokines to human organoids, and captured the effect of these cytokines. And we were able to show that there is some kind of overload in certain processes. And you can identify a profile of that, which very nicely resembles the biopsy transcriptomics, which contains many other cell types, indicating that actually organoids could be really good for mechanistic studies on cytokine interactions. The other input many epithelial cells are receiving is related to microbes. So our microbiolink tool is basically working with bacterial proteins, and we are working now on bacterial metabolite version of that. And we did a couple of studies on that, including one of my former postdocs did a really nice study on metaproteomics analysis. And basically, this is a very integrative approach where basically you have an input molecule and you have a transcriptomic profile of a cell. And our microbiolink approach can basically tell you the actual causation, the mechanistic connection between a transcriptomic profile and the input molecule. And then we also co-developed the OmniPath resource with Julio César Rodriguez's group, and that's really the big main resource that contains more than 100 different resources. If you would like to access anything easily, I would suggest to use that resource. It's good for cell-cell interactions and lots of other things, and it works with rodent models as well. And before I finish, I just wanted to mention a new venture I've got involved 1.5 years ago, and it could be relevant directly or just as a concept. So 1.5 years ago, we got the funding from NIHR, which is a major funder for translational medical research in the UK, to establish an organoid facility. And basically in this organoid facility, what we are trying to achieve is to have these kind of screening systems that I mentioned in the beginning of my talk, with low complexity, high throughput, but also disease models, which are more higher complexity, microbes, immune cells, and potentially lower throughput. And we use different reporter systems and genetic modifications to validate disease models. And we are trying to integrate these computational frameworks that my group has developed and made open access already, and combine these network biology and machine learning approaches for data interpretation. And we feel that it could be very useful for precision medicine. The key thing is that all of these pipelines are reusable and that they should be scalable, so they can be used for other projects. And then we can use organoids for testing molecules, cells, interactions, external molecules, microbes, and the goal is really to get to inter-organ connections. And we are working with engineers and industrial collaborators, including stem cell technologies, to increase the readout and the standardization, as I mentioned. It's quite important, in different materials. So it's a very, very exciting interdisciplinary story. And basically in this organoid facility, we're working with various tissues, not only gut. There's lung, brain, kidney, and other organoids, liver organoids. My colleagues are working. and we are supporting many new projects, and we are integrated in a really nice area of London with lots of different omics platforms. And we basically try to catalyze organoid research. So it's not a service providing facility. We are providing space, training, know-how, and industrial partnerships if needed. But basically what we want to achieve is that more people are using organoids, and ideally in their own lab, and they get more funding for that. They develop further these disease models, and they validate how they could work nicely. So just to finish off my talk, and this is kind of a summary how we use organoids. Previously we used basically mouse, as I mentioned, but now we are mostly working with patient-derived organoids. And then we create these very complex systems. We try to use them in a high throughput way. And then we are very keen on this kind of multi-omics and imaging-based readouts from these. And then we have these network resources I mentioned 5 minutes ago that basically we use to interpret the data to really understand the mechanism, what is happening there. And I didn't mention, but we have a really nice pipeline for patient genetic interaction information, which allows us actually to do patient-specific experimental samples and patient-specific genetic information to be combined, so we can create patient-specific models. And we hope that this could help to identify, for example, beneficial microbes, or understanding to prevent IBD, the onset of IBD or relapse. And the key thing with this pipeline is that it's a complex pipeline, but each module is hopefully quite clear how they work. And I think this is needed for a complex disease. We need to combine lots of different information for that. Okay, and with that, I just finish. I'm really grateful for lots of people currently working in my lab or used to work there in the last many years, and funders of our research, and lots of external collaborators who helped in these projects and working with us. And with this, I finish. I'm looking forward to your questions. Before that, I just wanted to mention that if you are interested about our projects, on our website, there are much more details on these projects and direct access to the papers. And if you're interested about the computational approaches and this GitHub link, most of our things are there, open access with documentation, so you can check it. And thank you very much.
Thank you very much, Tamas, for that fantastic talk and sharing your research with us. So we are now moving to the Q&A portion of this session. And if you see a comment or question submitted that you particularly like, feel free to give you a thumbs up, and I'll be looking at those questions and bringing them in during the Q&A. We have received a number of questions already, so I'll get started. So for your first question, can you describe the specific advantages of using the UMIX system compared to other microfluidic platforms for studying human microbiome interactions?
Yeah, that's a really good question. So I think one of the most relevant advantages of humics is that you can have anaerobic condition for bacteria, so you can keep bacteria that requires anaerobic conditions. And the other one is that the size of it is much larger than other gut-on-chip devices, which means that you can use it for multi-omics analysis. So you have enough material for parallel proteomics and transcriptomics analysis, for example.
Thank you. That's very interesting. So how do you address the potential limitation of using 2D organoid monolayers compared to 3D organoid structures in this system?
You mean in this system, you mean it was the question related to the humics system, or
Yes.
in general? So yeah, so basically the issue with the 2D monolayers is that on one side it's more expensive because first you need to have your 3D culture, and then you need to disrupt it and grow to the organoids. So it takes longer, it's more expensive. The second issue is that you need to have a confluent layer. So you need to double check it works like that. For example, in the recent version of the humic system, you have tier electrodes. You have electrodes to measure tier, so basically the confluency of this epithelial layer. And that could be challenging. So again, these are the points where I'm saying that we are still working, researchers are still working on making it useful. However, the complexity gain that we are getting from this system makes them much more useful than just working with 3D organoids.
Thank you, Tamas. We do have a question that popped up right at the end of your presentation. So it's the question is, does the approach of systems genomics that you mentioned earlier on can be compared with the network pharmacology at any level?
Oh, yes, absolutely. And my former postdoc, Dezső Módos, who is now a fellow in Imperial, is exactly working on this. So the good thing is that we can map the effect of the genetic background of a patient with our tools, and he's working on combining this with different footprinting approaches from cheminformatics and pharmacology. How looking at the different drug targets and different binding profiles, and how we can basically connect the two. So the drug footprinting and the genetic footprinting, and basically identifying which drug could work in which patient.
Thank you, Tamas. We do have some time to answer a few more questions, and our next question dives a little deeper into your methodology. So could you share some insights into the multiomics analysis techniques that you use to characterize the microbiome-driven changes in intestinal cells?
Yeah, so basically the key thing, sorry, the key thing is that first you are interested about what is there. So transcriptomics or single cell sequencing based transcriptomics is very useful for that kind of filtering a general signaling network, for example. And that helps, so you know that only those that are specific to that tissue or cell type is there, and you create your models. Then you can use, for example, phosphoproteomics to see the activation of different signaling pathways that you predicted that will be affected. And that's a really good filtering, as well as using different other specific markers to see whether it's really happening. So the first is really a mapping phase. The second is a validation type of approach. And it is essential for any computational model you do these because the data otherwise is very aggregated. So you need this kind of filtering and mapping. And you need the validation because in many cases, these predictions are just predictions and they are not always happening. And the last element of this work is comparison. So you compare between either healthy and disease, or having a genetic modification or not having it, because you corrected it with CRISPR, for example, and then you do a comparison. And this is where you are really validating your model, when you are specifically showing that, yes, this is the reason why the predicted output is there. And I have to say, it was only a very few times when we were able to go through all these three and reach the third one when we validated these. These were really great publications, and we're very proud of those. It's a very challenging thing. These are very complex systems, and even if we think we are right with the computational models, the organoids are not as cooperative as they should be often in the lab.
Thank you, Thomas. Our next question came earlier on during your earlier presentation. So beyond IBD, what other complex diseases do you see this approach being applicable to in the future? So are there any ongoing efforts to integrate different types of microbes or immune cells into the co-culture system to create a more comprehensive model of the gut environment?
Yes, so IBD is just one of the examples, maybe one of the most complex ones and challenging ones from that point of view. But there are many others. So from celiac disease to other diseases, including cystic fibrosis, for example, which severely affects the gut of patients, and these type of approaches are being set up to investigate these, as well as infection diseases. So we also did some studies with collaborators on, for example, on SARS-CoV-2 infection or salmonella infection using organoids. And this is really, really good to understand the role of the epithelium in these processes and how changes in immune cell epithelial interactions can affect these diseases. So absolutely, I think there's a plethora of diseases that can be and being investigated using organoids, and many of them have their own challenges compared to colorectal cancer-related organoids, which are a bit easier to grow, and the output is different. Basically, what you would like to see in these experiments. So yes, I think there are more and more diseases where it will be used.
Fantastic. Thank you for answering those questions. So we do have another question that was submitted very earlier on regarding the broader applicability of organoid models, which I think merits exploration, as I'm sure this is a question in everyone's mind are attending today. So the question is, how closely do these models truly mirror real pathology? So in your opinion, considering the complex human body, its intricate structures and cell communication, and the way disease unfolds over time, can we refine these models to be even more representative?
Yeah, it's a great question. Obviously, I'm here because my answer is yes. However, as I mentioned, there are lots of work to be done to reach that. I think it's a great start. I guess in 10, 15 years time, if we look back how we worked with organoids around these years, will be quite funny for the PhD students who are starting their work in 15 years time, because there will be lots of things solved and made easier and more realistic. I think the important thing is that the advancement of the field will come from engineers, and I don't think that biologists will be very important there, but engineering solutions, both in microfluidics, nanofabrication, biomaterials, will be the key for these closer to physiological modeling.
Fantastic. We do have some time to answer just one final question today. Um, yeah, so the question is, how do you introduce a protein from a bacteria? And by this, I mean, how do you discover the specific protein in regarding a microbiome?
Yeah, no, it's a great question. I didn't mention the details for that. So for the particular study, for example, that we did and published, we worked with a bacteria the Tetratetraomicron, with short BT, which produces extracellular vesicles. So basically, it sends out molecules, and then we isolated it with our collaborator and did a proteomic study on them. So that's a really good way, actually, to capture molecules that are being used for communication purposes. The other option is just to having a bacterial culture and use the media secreted molecules from there that you suspect that would reach a cell, a host human cell. And basically, we are doing this, for example, with metabolomics. And then we identify these metabolites. Metabolomics is much more narrowed than proteomics from that point of view. But you could identify molecules with this, and then you need to validate that. So you need to adjust that protein, for example, or in our project, we did some mutations of the bacterial strain to remove the protein. So we had the very same vesicles, but that particular protein was not in it. And then we compared the results for that.
Thank you so much, Tamas. Yeah, we do have a question that just came in just a second ago. I think we have just a minute to answer this
Okay.
question quick. So, is it possible to use genome-edited stem cells in organoid-based system to check any off-target effect prior to transplantation?
Yeah, so yeah, we haven't really talked about transplantation of organoids back to patients, which is, I think, another cool area. But yes, I think yes. The short answer requires lots of research first. What we are modifying there and what the effect for that for transplantation, for example. But definitely, I think that there's a big power on genetic modification of organoids.
Thank you so much, Tamas. Yeah, unfortunately, that is all the time we have for today. Any questions that we didn't have time to get to will be sent to the speaker after the event. And as with all the other talks in the event, this talk will now be available to view whenever you want for the next 30 days. That just leaves me to say thanks again to Dr. Tamas Korcsmaros, and to all our viewers. We'll be back soon with the next talk.
Thank you.